Mark Krieger. I'm from the Dartmouth-Hitchcock Medical Center and the Geisel School of Medicine at Dartmouth in New Hampshire. And I had the honor of being a director of one of the American Heart Association's vascular disease strategically focused research network. This network comprises 4 centers, and you'll be hearing from my colleagues who directed other centers of our network. The network was really focusing on vascular diseases, and 2 in particular, peripheral artery disease and aortic aneurysms. And it gave all of us the opportunity to really think about the science underlying these vascular diseases so that we can further understand the mechanisms which will at least enable us to consider and initiate treatment interventions which will improve the outcomes of patients with these vascular diseases. So our SFRN really had to do with 2 important vascular diseases, peripheral artery disease and aortic disease, each of which are very prevalent in our country and actually throughout the world. It's a problem encountered by many individuals, contributing to their morbidity, and unfortunately also is associated with increased mortality in each circumstance. So the SFRN, which we discussed today, really involved the work that was done at each of our centers, and you'll hear from the other speakers in just a moment, talking about the work we have done as individual centers, but importantly the work we have done in a collaborative way to really enhance our understanding of vascular diseases so that we can identify the problems that patients encounter and hopefully develop new treatment strategies to improve their quality of life and also their long-term survival. I, for one, was one of 2 directors of the Brigham and Women's Hospital Dartmouth-Hitchcock SFRN Center, and our focus was on peripheral artery disease and diabetes and its manifestations in critical limb ischemia. Critical limb ischemia is when the leg is threatened because of a severe impairment of blood supply, and many of these patients are at risk for amputation. And since in each case, both with diabetes and with peripheral artery disease, there's a large association with systemic atherosclerosis, they're at risk for other cardiovascular events such as heart attack, stroke, and death. So our center had 3 components to it. It had a basic research component, a clinical research component, and a population component. Our basic research component was really looking at the work with RNA and specifically non-coding RNA and how it may affect preclinical models and ultimately patients who developed critical limb ischemia as a consequence of both peripheral artery disease and diabetes. Our clinical research problem was actually looking at clinical markers as well as biomarkers that may predict adverse events in patients who have peripheral artery disease and diabetes, and by so doing maybe identify appropriate targets for therapy. And for that we used a very large clinical database of over 31,000 patients based on 11 clinical trials that have been conducted in different areas, particularly at Brigham and Women's Hospital as part of the TIMI group and also overseen by the University of Colorado. And our population study was really looking at how disparities in care are really affecting the prevalence of peripheral artery disease in populations throughout the country, and how limited access to care and really reduced attention to preventive measures is affecting outcomes. As a whole, putting all these things together, we've really taken it from the bench to the bedside to the population to really improve our understanding of the mechanisms of peripheral artery disease and diabetes, its manifestations, how these manifestations predict prognosis, and then allowing us to really focus on individual targeted therapies and population therapies to improve outcomes in this disease.
Okay, I'm Alan Doherty. I'm at the University of Kentucky. I had the pleasure of leading a group that was both a collaborative project between the University of Kentucky and now very close colleagues at Baylor College of Medicine. Our particular disease focus was on aortic disease, with a particular emphasis on thoracic disease. There is a growing awareness that these diseases are very prevalent, and when detected, unfortunately, only surgical options are available to the afflicted individuals. So there's a great need for background information on these diseases to really give some insight into potential targets for drug therapy. So we, like the other centers, had three particular projects. We had two that were mostly focused on basic science. And the drift of these basic science projects was really, what is the role of sex that has in this particular disease process, and how does that sex influence both the inflammatory components with different neutrophils and macrophages that would come into the diseased tissue, and also the matrix which gives the stability to the tissue, how is that affected and how can it be regenerated? And then we had this great interaction with the Baylor crowd, in which they have a phenomenal access to many, many clinical samples. They have a tissue bank of 4,000 aortas. They do 400 procedures a year. It really gave us an amazing access to samples to really pull these away and sort of dissect out the same mechanistic basis on human tissue. I would say also probably one of the bigger benefits we have of this great collaboration was that the Baylor group has deep expertise in a lot of genomic analysis. And so this really gave us all a great insight in between able to expand our research efforts during this 4 years of the support to really give vast new insight with a real focus on, what could we do about some of these components that we could potentially build a therapeutic. And that's what we were discussing at this session about therapeutic options that we think hopefully will be applicable to humans down the road.
My name is Josh Beckman, and I was lucky enough to be the center director for the Vanderbilt site of the Strategically Focused Research Network in Vascular Disease. One of the more amazing things that you'll see amongst the four of us is that there were many complementary projects. The first project that you heard Dr. Krieger describe looked at the overlap between peripheral artery disease and diabetes in severe limb ischemia. We were quite interested in trying to understand why patients develop symptoms with peripheral artery disease. Our focus was on the microvessels and how the microvessels impacted how people feel. Our basic project used intravital microscopy to study live insulin-resistant mice and understand the different ways by which hormones to bring glucose into tissues and glucose itself were able to translocate from the small vessels into the tissues. What we found was that time across the interstitium was probably the more important determinant of whether or not insulin could get the glucose in, and the rate at which glucose got into the tissue in the small vessels determined its availability. This seemed to be a process that was interrupted or at least inhibited by inflammation, and when there were, and it's an integrin-dependent process. We then can take a look at our population studies, and we looked again at microvessels, but this time microvascular disease. What we're able to say now is that the presence of microvascular disease amplifies the risk of peripheral artery disease, such that when you have the two of them together, compared to people who don't have either of them, the risk of having an amputation is about 25-fold higher. This is a giant risk and really tells us who are the patients at risk that we have to pay quite close attention to. We've done other work in this area trying to figure out who gets the disease, peripheral artery disease, finding that blacks and whites seem to get the disease at similar rates, although they don't have similar outcomes, suggesting that health access may be an important part of determinants. And then we tried to look at using translational studies at why patients don't feel well. And we have found that microvascular disease in the skeletal muscle is a strong determinant of how far people can walk and likely participates importantly in how people feel. And we've also found that there's a change in metabolism such that there's a move towards branched-chain amino acids away from more of the standard metabolites, suggesting that the change in muscle over time is being affected by the different nutrients that are available, both for energy utilization and for growth and repair. And so the work overall has really brought together the idea that microvessels and the local tissue participate as importantly as the large vessel disease and peripheral artery disease. The last thing I'll say is that all of this work came together because we had a relatively common vision among a disparate group of scientists, and the coming together made us all smarter for the efforts.
My name is Mary McDermott, and I was the Center Director for the Northwestern University SFRN on vascular disease. And our projects focused on skeletal muscle abnormalities in patients with peripheral artery disease. And while prior science had demonstrated muscle abnormalities in people with PAD who had chronic limb-threatening ischemia, our work focused on patients with PAD who had more mild PAD, such as, for example, intermittent claudication. And we had a basic science study and a longitudinal observational study that evaluated the association of the ankle brachial index with mitochondrial activity and also mitochondrial proteins. And in our basic science study, and consistently found in our observational longitudinal study, we found that patients who had mild peripheral artery disease had lower levels of mitochondrial proteins and also lower mitochondrial respirometry, while those with severe PAD had almost a compensatory response. And the overall finding, therefore, was that patients with peripheral artery disease had respirometry activity that was similar to those without PAD, but one had to look at the ankle brachial index to notice that actually across the range of ABI, those with mild ischemia seemed to have a decrement, and those with more severe PAD seemed to have a compensation in terms of their mitochondrial activity. That was a surprising finding for us. Also of interest was that those without peripheral artery disease had a strong direct association between the ankle brachial index and mitochondrial activity, suggesting that even among people who have a normal ABI and don't have peripheral artery disease, higher ABI values may still be better. And we also found that in those without peripheral artery disease, there was a direct correlation between 6-minute walk and mitochondrial activity, but in those with PAD, we saw no such association, which was also surprising to us. We also conducted a phase 2 randomized clinical trial of nicotinamide riboside with and without resveratrol. This is a double-blinded, placebo-controlled trial of 90 participants. And I presented preliminary results today suggesting that there was a signal of benefit of nicotinamide riboside for improving 6-minute walk distance and treadmill walking distance in patients with peripheral artery disease, although the addition of resveratrol did not improve the effects of nicotinamide riboside compared to placebo. So overall, in the 4 years, we felt that we made tremendous progress in the study of the importance of skeletal muscle health to walking performance in patients with peripheral artery disease, and also had the chance to collaborate with the other 3 centers to start some new projects that we look forward to carrying forward as well.
So as you can see, this was really a wonderful opportunity for 4 centers coming together with common themes to study vascular diseases, peripheral artery disease at 3 of the centers and aortic disease at the 4th. But importantly, it gave us the chance to work closer together, share our science, and really, through that collaboration, enhance the entire field. It really underscores the importance of the American Heart Association's strategically focused research networks, really accomplishing what it's supposed to do, which is to not only conduct research, but do it in a synergistic method so that we can move fields forward. And I think all of us together have done that in the field of vascular disease.